Fine. Well, thank you very much. I think this is going to be a very stimulating afternoon. And as my title indicates, I shall be giving you an idea of some of the approaches we are using to investigate whether there are any epigenetic consequences in the egg of maternal diet. And just as a spoiler alert, I should say that some of what I present this afternoon is very much work in progress, so I shall not be able to give you a final answer to the question posed in the title. Sorry for that disappointment. So first of all, I think it would be useful to remind ourselves that the egg or the oocyte passes on far more than just the genetic material to the next generation. In addition, it contributes all the machinery for the resumption of meiosis and cell division, for the orderly activation of gene expression in the embryo, the energy sources to power up the embryo, and much more besides. In addition, it does provide some essential epigenetic information. And of course, it's possible that any or indeed all of the above can be altered or impaired to some degree by adverse environmental exposures or adverse physiological states, for example, maternal obesity. And these collectively could contribute to impairments in fertility. We are particularly interested in epigenetic effects. I think they certainly deserve to be investigated. And that is primarily because they have the possibility of inducing longer term or later onset effects in the offspring. So of the various epigenetic modifications that exist, and I'll remind you of that scheme that Stefan showed us. Of these, we know that DNA methylation is an essential mediator of epigenetic transmission between the generations. And this is the case for genomic imprinting. And this is a completely normal mode of epigenetic inheritance in mammals. So we know that imprinted genes, of which there are a couple of hundred, are expressed from only one allele or copy in the offspring. And that is because these genes have been marked in different ways by DNA methylation in the sperm or the egg. And so in this example, the ZAC or PLAGA1 gene is expressed only from the father's copy because the mother's copy is silenced by DNA methylation that was imposed in the egg. And we know that this is important because without this methylation, the gene becomes aberrantly overexpressed, leading to severe intrauterine growth restriction of the fetus, absence of insulin production, and resulting in a syndrome called transient neonatal diabetes. So imprinted genes, as I'm sure many of you are aware, are the best example of the importance that DNA methylation states in the egg can have for the next generation. But it's possible that the influence of DNA methylation is more widespread than just this select number of genes. So I'd just like to make a brief detour to give you a little bit of context, which will help interpret much of what I'm going to say in the next few slides. And this is to show you that the genomic pattern of DNA methylation in the egg is rather unusual, in that methylation is present only over expressed genes, as you can see in this profile. And if we focus on a single imprinted gene, this is a ZAC1 gene now in the mouse, we can see that the pattern of methylation very neatly covers the extent of the expressed gene. And if we prevent the gene from being expressed by deleting its promoter, then the gene will not become methylated in the oocyte. And this is important because it leads to the prediction that If out of some physiological or pathological circumstance there are gene expression changes in the oocyte, then there should be corresponding changes in the DNA methylation pattern with the potential for inheritance. Of course, removing a gene promoter, as we've done here, is obviously a very dramatic and artificial act. But can we see evidence to support this prediction in more physiological settings? So just look at this study briefly. This is a study that we've done in collaboration with a group of Johan Schmitz and Ellen Ankert in Brussels. And they were interested to evaluate whether some of the procedures commonly employed in assisted reproduction could alter the methylation pattern of oocytes. In particular, oocytes obtained by in vitro culture and in vitro maturation, or after hormone priming and superovulation. This study was done in the mouse as a model, but the group is clearly interested in applying this in human practice as well. So in the study, we performed whole genome methylation profiling of pools of oocytes obtained after the following 4 protocols, either in vitro follicle culture, or superovulation of age-matched young females, or superovulation of adult females, or completely naturally ovulated oocytes. And reassuringly and broadly speaking, the methylation profiles of all 4 groups were very similar. But if you look at this PCA plot here on the right-hand side, you could see that the groups do separate, indicating that there must be some consistent reproducible difference in the methylation pattern according to the treatments. And indeed, we can find examples of whole blocks of DNA methylation that differ between the groups, in this case associated with the expression of the gene TCF4, in this case between oocytes obtained from young and older females. So we do find evidence of altered methylation patterns downstream of altered transcriptional states in the oocyte. Okay, so now let's return to the question posed in the first slide. So this is a study being conducted by a Marie Skłodowska-Curie postdoc fellow in the group, Antonio Galvão, and it's done in conjunction with his group in Olsztyn in Poland. And he's using a very well-characterized model of diet-induced obesity in the mouse, where you subject females to 3 or 4 months worth of high-fat diet feeding. And he is assessing the effects of the diet on the gene expression and DNA methylation in oocytes, in embryos at the onset of zygotic genome activation, in blastocysts before implantation, and in early post-implantation embryos, because he wants to get a comprehensive account of the effects that might have occurred in the oocytes and whether they persist into the next generation. So first, however, I thought I would show you this interesting finding where he examined gene expression in cumulus cells. So these are the companion cells of the oocyte throughout its development, and they help to nurture it and are expelled together with the oocyte at ovulation. And he's been looking at these cells by RNA sequencing. So what he finds is quite remarkable. In the plot on the left-hand side, he is displaying a measure of the variation in gene expression between the different groups, depending upon the length of diet and whether the females are gaining weight or not on high-fat diet. And he says, and he sees that the major variable that accounts for almost 50% of the variation you see in gene expression is directly proportional to the body weight of the female. And you can see a very similar thing on the heat map on the right-hand side, where 800 or so differentially expressed genes are plotted where you see there's a very linear and quantitative effects on the expression of these genes according to the weight that the female mice have attained. So the companion cells of the oocyte that are helping to nurture it during its development show a very marked transcriptional response to the abnormal physiology associated with obesity. Okay, so let's move on to the oocytes. So for this, we really need to invoke single cell approaches. And that is because there are far fewer cells available to analyze. We're also interested to see whether cells are responding in the same way or whether different oocytes respond differently. And ideally, we would like to have a method that enables us to take multiple different measures from the same cell. For example, because as I showed you before, DNA methylation and transcription are very much coupled in the oocyte. This slide just illustrates that we can obtain very good representations of the genome-wide pattern of DNA methylation even in single oocytes by using a modified method for bisulfite sequencing, although the data are rather sparse in comparison to bulk populations. And furthermore, in collaboration with other groups at Babraham at the Sanger Institute, we have been able to obtain parallel profiling of DNA methylation and gene expression from the same cells. Okay, so moving on to the diet study. So Antonio was able to obtain up to about 70 ovulated oocytes per group from 6 to 7 female mice, and these were processed for single cell methylation profiling or RNA sequencing. So first of all, if we look at the methylation, when we merged the data sets by group to get a very comprehensive picture of the DNA methylation, then broadly speaking and reassuringly, the bimodal pattern of methylation characteristic of the oocyte is preserved irrespective of the diet. However, that's not to say that we cannot find differences, and the scatter plot on the left-hand side identifies over 400 sites where there's a significant departure in methylation in the high-fat diet compared with the control diet counterparts. And in some cases, these sites of methylation cluster together and gang up across whole genes. And on the right-hand side here, you see a single locus, the GLY3 gene, for which there's a very consistent difference in methylation across the whole of the locus, comparing the high-fat diet and the control diet's oocytes. And again, because this occupies a whole gene body, we would suspect that that would be downstream of a transcriptional effect. So analysis of the same oocytes by the single-cell RNA sequencing again reveals a globally preserved gene expression profile, but there's a tendency for the oocytes from the high-fat diet females to show greater variation in their gene expression profile. We can identify differentially expressed genes, and the heat map on the left-hand side identifies the top 300 or so of those, which you can see will partially partition the cells and the oocytes according to their diet group. And these differentially expressed genes will show some enrichment for a variety of GO terms or processes, such as cellular metabolism or cell cycle control. And going back to the coupling between gene expression and DNA methylation, for a small number of genes, we can find a definitive correlation, positive correlation between the change in transcription and the change in DNA methylation observed. So, as I said at the outset, this is very much work in progress. We've still got a huge amount more to do in analyzing the data sets from all these stages. We have been able to observe effects on the methylome in oocytes associated with high-fat diet and obesity. We would like to be able to explain them. And as a molecular biologist, we want to have an explanation rather than to record them as random, inexplicable events, and trying to find rational explanations, for example, through transcriptional changes, will be reassuring to us. We want now to follow these through to the embryonic stages to see to what extent the changes persist or whether new changes arise as a consequence of some other impairment that the diet has done to the oocyte. And we want to be able to infer how many of the changes would indeed be functional, i.e. could impact the expression or the potential for expression of genes in the next generation. So that remains to acknowledge the people who have been involved. I indicated Antonio Galvao as the principal mover of the diet study. I'd also like to highlight Juan Casero Fernandez, the bioinformatician within the group, for some very critical inputs, as well as the funders. And I think at that point my time is up and I'm happy to take questions.